Okay, we're going to continue with the second video on Chapter 16, Cell Signaling. And we are going to continue with general principles of cell signaling and focus on the types of proteins and activities happening as a signal is received and then converted to a change in cellular behavior. In the first video, we looked a bit at extracellular signaling molecules and receptor proteins. Here we're going to continue on to look at how the message that's received by the receptor protein at the extracellular surface is converted into a change in behavior in the inside of the cell. Binding of an extracellular signaling molecule will trigger conformational change that allows that receptor to activate proteins within the cell. Okay, so we've got this kind of listed here. The extracellular signaling molecule, it's activating the receptor. The receptor receives the signal and passes the signal to the cell interior. This is sometimes called transduction, conveying the signal from an exterior signal to an interior signal. In this case, they're showing us three intermediate proteins of signaling molecules where one activates the next, activates the next. So none of these green proteins are directly changing cell behavior. They're passing the signal along. By the time we get to the blue proteins, we're thinking about effector proteins. So this is the protein that's actually going to make a change within the cell. So a metabolic enzyme, that's the effector, the protein that's making the change, and the effect, or targeted cell response, is altered metabolism. In this case, the effector protein is a cytoskeletal protein. So this might be involved in changing the shape, so producing a neuron, making a dendrite, making an axon, or a cell moving itself. We're going to see a number of examples where what we're activating is a transcription regulator, and this will mean producing new gene expression. And remember, this would be a longer-term effect because we have to wait for transcription and translation to occur. So I've posed the question here, why are cell signaling pathways so complicated? And the answer to that isn't easy in itself, but one way I think about it is that the complexities of the signaling pathways gives us opportunities to regulate the cell, to kind of fine-tune things. And here are some examples of things that can happen when you have a more complicated signaling pathway. So in the first case, begin again with an extracellular signaling molecule, and the receptor is doing something called primary transduction, changing the extracellular signal into an intracellular signal. And it happens to be that this receptor protein is forming a protein complex. So this is the scaffold protein that's sticking to the receptor, and these two proteins in the pathway that are the next to be activated. So the receptor activates this protein here, which then relays or passes on the information to an enzyme, most likely, that's going to amplify the signal. So for every one extracellular signal molecule that's activating the receptor, relay the message, and then activate an enzyme, this enzyme can make large quantities of small intracellular signaling molecules, which can then activate many targets within the cell. So this is an amplification step because we're creating many of these individual intracellular signaling molecules that can spread throughout the cell. And also it's a transduction because we're changing the form of the cell message again. Okay, so the enzyme can produce large numbers of what they're calling small intracellular messenger molecules. A common name for them is also second messengers. So I've just added this text. Second messengers is another term for these small intracellular messenger molecules, right? The extracellular signal would be the primary or the first message, and these intracellular signals are second messengers. So that's one of the things that can happen with these more complex pathways is having a second messenger that can amplify or produce a large response in the cell for every one signaling molecule. We can also see integration happening here. The particular second messenger can bind to and activate this odd-shaped protein. But it looks like in this picture here that this protein and two others are actually required to activate this triangle protein. So these proteins, this odd olive green shaded protein, is going to be activated by a different cell signaling pathway, and this triangular light green is activated by another pathway. All three of these pathways must be active to turn this particular protein on. So this is integrating. So multiple extracellular signaling pathways, all of them have to be active at the same time for a single response. Another mechanism for integration is the example of the formation of a complex, where maybe these three proteins, instead of all activating the triangle protein, maybe those three proteins need to come together and form something new, all made of all three pieces, to continue on the signal. That is integration. And then we can see that this protein turns on, the triangular protein turns on this protein. You can see it binds to it. And this is one before the effector protein. In this case, this particular protein is going to distribute the message by turning on multiple effectors. So one signaling molecule here, this one that binds to the triangle, is going to turn on an effector that alters metabolism and also turn on a effector that alters cell shape and or movement, and also turn on a transcriptional regulator that alters gene expression. So all of these effects are going to come from one protein. I'm going to note, I don't remember if I said it earlier, but a common way of describing the steps in a pathway is that the earliest effects are upstream in the pathway, and as we move down the pathway, we're going downstream. So this enzyme is downstream from the receptor and upstream of this signaling protein. So another thing that can happen is feedback. This feedback is still affecting activity of proteins. I say still because we looked at this in biosynthetic pathways in chapter 4. This is not a biosynthetic pathway. We're not making a product necessarily. The steps is relaying the message or kind of passing the message on from one to the other. But the idea of feedback is similar. So this protein here that is turning or causing these changes in cell behavior can also affect its own activation. So not its production, but its activation. So that's what's different. I'm just noting that feedback in a cellular signaling pathway can also be positive feedback or negative feedback. Positive feedback can be used when you're trying to have a really rapid response in a cell. There would be another mechanism for turning it off, but if you have a protein that activates itself, you can have a very rapid response to one signal. We see positive feedback happening in the cell cycle, which you're going to study in the next section. You can also have negative feedback where the protein that is activated shuts down its own activation. This is also seen in the cell cycles when we want to shut down a pathway after a certain amount of time. And this is a reminder of regulators of protein activity that we already learned about in chapter 4. Kinases, phosphatases, and G proteins are common components of cell signaling pathways. So remember that a protein kinase phosphorylates proteins. This can turn them on. It does not have to be an activator. Protein phosphatases remove phosphate groups. Basically, a protein kinase and a protein phosphatase toggles or switches a protein between two conformations, either of which could be the active. The phosphorylated form could be the active, or the non-phosphorylated form can be the active. It is going to be that most of the cases we will see in this chapter, they are showing you situations where phosphorylation is activating the protein. We also covered G proteins. Remember, G proteins do not covalently bind to GDP or GTP, but they have a binding site that can fit either. If we start with this off conformation where the protein is bound tightly to GDP, we first need to remove the GDP, make it leave this binding site. Some type of signal will probably regulate this. The signal will turn on a guanine nucleotide exchange factor, or GEF. At that point, GTP can fill in the space, bind to the site, because it's at a high concentration within the cytoplasm. The G protein will be active and then can continue on the signal until the protein hydrolyzes the last phosphate off of GTP and converts it to GDP. And again, the G protein will be off. Later in the chapter, we'll look at two cell signaling pathways, or two common families. One is receptor tyrosine kinases. Those receptors turn on many kinases and also at least one G protein. That is a simple G protein like this. And we're also going to look at G protein coupled receptors, which activate G proteins, but they're G proteins that are usually found in a complex with two other proteins. So we'll look at those examples a little later in this chapter. The textbook introduces three broad families of receptors, or types of receptors, that are very commonly used in cells. The first one is an ion-coupled receptor that we've really already covered when we were looking at neurons, right? So this would be an ion channel that is in a closed conformation. Here are the ions that could normally pass through the channel. And then we have the signal molecule. In the case we looked at of neurons, this was a neurotransmitter where binding causes a conformational change, opening the channel, and the ions can pass through. I'm going to finish up giving you a broad overview of G protein coupled receptors and enzyme coupled receptors, and in the next lectures we'll go into quite a bit of specifics on those two pathways. But to begin, what makes a G protein coupled receptor, which is here in green, is the fact that it interacts with a trimeric G protein. So here I have added in the labels that you'll see pretty much for the rest of the slides that there are three subunits, an alpha, a beta, and a gamma. The alpha and the gamma have a lipid link that keeps them tethered to the membrane. They're kind of stuck right at the membrane. And when this G protein coupled receptor is activated, it will have a conformational change, which you can see going from right here in this intracellular domain, that allows it to interact with this trimeric G protein, the alpha, beta, gamma subunits, and this alters their activity. They're not showing it here, but the alpha subunit, when it's complexed with beta, gamma, is bound to GDP. The alpha subunit is what defines these as a G protein, right? Because G proteins bind to guanine nucleotides, GDP and GTP. When the alpha subunit is combined with beta gamma, it should be bound to GDP, and I'll show that in the later pictures. Binding of the signal molecule to the G protein coupled receptor causes alpha subunit to dissociate from beta gamma, and that allows it to release the GDP and bind GTP. So in the inactive state, alpha, beta, gamma, one complex, bind the signal molecule to the G protein coupled receptor. We separate the alpha from the beta gamma, and the alpha is activated. And again, both of these can relay signals. Okay, types of receptors. Again, the third type we're looking at are enzyme coupled receptors. These are different in that the receptor isn't kind of fully formed until it binds to the signal. So there are two cases. You can see there's kind of an OR in the middle here, where we have two subunits that will come together to make an active receptor. This is a homodimer, meaning the two proteins that come together have the same sequence of amino acids. When there is no signal present, they're not next to each other, they're not close. The signal itself is a dimer. You can see it has kind of a repeated shape. They've shown you one in a pinkish color and one in red. And when the two subunits, the identical subunits, bind the dimer signal molecule, it brings them together. And the two subunits will become active. And the receptor subunits are themselves enzymes. We will look at this in the specific example of receptor tyrosine kinases. So their enzymatic activity in the cytoplasmic side is a kinase activity, so they can phosphorylate. So we're going to look at that kind of generally in the textbook and then more specifically in the case study on insulin signaling. Enzyme-coupled receptors can also be heterodimers, where the two proteins coming together do not have the same sequence. And here they're showing it as a monomer. Binding of the signal still requires that these two subunits of the receptor come together. And this is a heterodimer because they're different from one another. And in this case, rather than having an enzymatic activity that is inherent to the receptor then activates an associated enzyme. I'll end our video here and we'll move on to the more specific study of these two pathways in the next lecture.